So Frederick is from a life science background. He has done BSc in biomedical science and MSc in biotechnology. So we have a lot of life science students amongst us. And all of them are in a beginner level, except Frederick. So do you have any particular interest in such as oncology or infectious diseases? What are your research interests? I'm curious to know about that too. Some people are interested in spaceomics. Some people are interested in machine learning. What are your research interests? All right, so we have a diverse range of research interest amongst the participants. So Abhimanyu says he is interested in neurodegenerative diseases and oncology. And Kursit says he is interested in omics data analyzing in inflammation and inflammatory disease. Varsha is interested in clinical research and has also worked in medicine research projects and is still exploring research projects. That's a pretty cool, diverse range of interest amongst the participants. I'm curious to know about rest of your interest as well. All right, so while we are waiting for the participants to share their interest, I'll be starting with today's webinar. So hello everyone, a very warm greeting to one and all. My name is Srigauri Krishnakumar and I would like to welcome you all for today's webinar on Introduction to Bioinformatics, Multi-Omics. This is an online program and is designed for everyone, including students who don't have a background in bioinformatics and biologists to learn about next generation sequencing data analysis, including genomics, transcriptomics, and metagenomics. As a result, participants will become proficient in the analysis and interpretation of various types of omics data using bioinformatics, including commonly used statistical and machine learning techniques. And in today's webinar, we will first sign up on the Omics Logic Learn portal and learn how to navigate the various courses that will be part of your program. Followed by this, we will go through the details of the program, such as topics covered in each session, associated coursework, and example projects. Finally, we will cover the details about the program registration process and program cost. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who's working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the Omics Logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Students will also have access to the omics logic resources. But how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This also comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics that includes genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Users will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. Different stages of analysis are performed in different sections on this Multi-Omics platform. Another important resource that you will get access to as a part of the program is access to the Omics Logic Code Playground, which will help you develop coding skills while learning popular packages in R and Python to visualize, annotate, and analyze complex patterns in biological data. And with so much data available on any biological topic and question, completing a program seems like a great opportunity, but it can be challenging. That is why you also have the opportunity to seek mentor guidance and learn alongside an international community of bioinformatics and data science enthusiasts. So now, to view the various courses, you need to first sign up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, a brief bio about yourself that will consist of your educational background and research interests. The mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the Omics Logic Learn portal. So I'm sharing the link in the chat box for the participants to sign up on the portal. So here's a brief demonstration on how to sign up on the portal. So for those participants who do not have an account on the portal yet, you can simply click on the Create an Account option that you see here. So once you click on this, you can fill out your name, email ID, and password, or you also have the option to sign up using any of your social media accounts, such as Google, Facebook, Apple account, GitHub account, or even Twitter. And for those participants who already have an account on the portal, you can simply click on the Login Now option that you see here and simply click on Sign In. And with that, I would like to pause for a couple of seconds for the participants to sign up on the portal. And if you already have an account on the portal, please put a yes in the chat box. And once you have signed up on the portal, please put a done in the chat box, and I'll proceed along how to show the various courses and example projects. Has everyone visited the link? And if you're having any queries, you can mention in the chat box as well, and I'll be happy to help you. So we have one yes from Abhimanyu. I'm expecting responses from the rest of us as well, because I'll be sharing links to the coursework and it'll be easy for you to open the link and navigate through the various lessons in the coursework. For that, you need to have an account. So Varsha says she's already registered. What about rest of the participants? Have everyone signed up? So I'm going to sign into the portal, but along the way, if you're having any queries, please do let me know. So once you sign in on the portal, you'll be able to see the various courses and example projects. So in the search bar that says looking for a specific course, if you have a specific research interest, you can simply enter that keyword. For example, let's say you're interested in transcriptomics. So once you enter that keyword, it will show up the various courses and example projects that make use of that concept. And you can simply navigate through and browse through and take a look at all of the courses and example projects that are there. But for now, coming to the part where I was talking about updating your account on the portal. For that, click on the tab that says Welcome Back with your email ID. So once you click here, to update your account, you can simply click on the button that says Profile. So once you click on this, you will be able to edit all of your details that I explained in my presentation. And this is the activity tab where the community manager and the mentor for your program will be tracking your learning progress. And the courses you are currently completing will appear under this course tab. So once you complete a project and upload it on the portal, that will be displayed under the projects tab. And once you enroll for any of the programs, such as the Introduction to Bioinformatics, Multi-Omics, then that will be appearing under the Programs tab. So now let me show you through the sessions of this program. So you can simply enter, when you click on the tab that says Programs and enter the Introduction to Bioinformatics, it will show up the program that you're interested. So if you navigate down below, you'll be able to see the various sessions that will be there as part of the program. I'm sharing the link in the chat box so that the participants can have a look at it. So now going back to my presentation, we will take a look at each of what are these sessions all about. So we know that a digital revolution is happening in healthcare, and humans are being digitized through new devices, apps, and monitoring technologies, which are tracking, analyzing, and storing large amounts of data. And for many diseases, molecular data is increasingly important and relies on complex bioinformatic analysis. And for many wet lab scientists, the analysis of large amount of data is still a major bottleneck. And against this backdrop of this fast-moving field, to address the challenges for bioinformatic education and training, we have designed this program on Introduction to Bioinformatics, Multi-Omics. So now let us go over the sessions one by one and take a look at the associated resources and the session outcome. So the very first session will be on bioinformatics and big data. Here we will introduce the participants to the various fields of bioinformatics, important concepts, and its various applications in biomedical research. And for this purpose, we will be going through a few introductory courses. So first of all, you'll be asked to complete course 1, Introduction to Bioinformatics, where you will cover topics on big data bioinformatics and its uses in basic research, healthcare, and the biotechnology and pharmaceutical industries. And at the end of the course, you will have a thorough understanding of what is big data biology, what are various applications of bioinformatics in healthcare, and what is translational bioinformatics. And another interesting course that you will be taking as part of the program is course 2, Bytes and Molecules. Here you will gain an understanding of how the current revolution in biomedical data impacts biomedical research and human health. And at the end of this course, you will be introduced to various omics fields, including genomics, metagenomics, and transcriptomics. But so far, we have been gaining an introductory level knowledge of various omics fields. So now we will go one step further and explore each of these fields in detail. So now, let's say a sick patient comes to you with their blood or tissue sample, and you want to understand what genes are affected, what is contributing to his or her diseases. For this, we need to carry out genomic data analysis. So for this purpose, we have the second session that will introduce the participants to genomics, where you will learn about DNA variants and mutations. And other topics covered during the session include germline and somatic mutations, analysis using mapping, detecting variants, and interpretation of significant and insignificant mutations. So now I'd like to take you through the courses that will be taught as a part of the session. This will be course 3 Genomics. And in this lesson, you will be familiarized with the biology of genetics and genetic variation while considering practical applications. And at the end of the session, you will learn about variant calling analysis to process data and identify variants between cancerous and non-cancerous cell genomes. The full course also contains other important concepts such as phylogenetic analysis, mutability analysis, differential mutability analysis, and so on. So another important session is on metagenomics. So now I'd like to ask you all a question. So do microbial communities have a role in contributing to diseases? The answer is yes. For instance, a recent study has found that the appearances of Alzheimer's disease has been tightly associated with changes in the skin microbiota. And with the pace at which metagenomics data is generated recently is astonishing. So that is why we have designed this session for metagenomics. And there will be 2 sessions for this. First is designed to introduce the participants to the various fields of metagenomics. And the second session is for introducing the participants to metagenomic sequencing and its various applications. And as a part of this session, you will be directed to course 4, Metagenomics, wherein you will learn to analyze microbiome composition and species diversity using metagenomics data. And at the end of the session, you will gain a basic understanding of data2 or KIME2 analysis of amplicon sequencing. You will also learn to interpret the outputs, including alpha diversity, beta diversity, and even taxonomic abundance plots to understand the evolutionary trend through phylogenetic analysis. So now another interesting question is it possible to find patterns in the gene expression between a healthy and a sick patient? And if so, how can we do that? For this purpose, we'll be making use of transcriptomics data. For example, the image displayed in front of you is part of a study wherein the researchers try to compare gene expression changes in the immune responses before and during malaria infection in the patients. But how do we analyze it? So for this purpose, we have designed 2 more sessions on transcriptomic data analysis. In the first session, we will learn about gene expression data analysis. Followed by this, in the next session, you will learn about differential gene expression analysis. And as a part of this session, you will be directed to course 5 transcriptomics, wherein you will learn how the same genome can give rise to different cell types and how gene expression is regulated. And at the end of the session, you will have a thorough understanding about transcriptomics data analysis, downstream analysis of transcriptomics data, from basic visualization to statistical analysis of differentially expressed genes, all done by using the popular DSEC2 package. And you will also learn about biological interpretation and annotation of gene sets. So the above 2 sessions were solely focused on bulk RNA sequencing, wherein you will be looking at the gene expression at a large scale. But is it possible to identify gene expression pattern of individual cell, let's say in a tumor ecosystem? The answer is yes. Unlike the conventional bulk RNA sequencing methods, the single-cell RNA sequencing provides an unbiased insight into the gene expression pattern of each individual cell, thereby providing a comprehensive look into the mechanisms of cell-to-cell interactions. And to help you gain these skills, we have designed the Introduction to Single-Cell Transcriptomics session. And as a part of this session, you will be directed to course 6, Single-Cell RNA Transcriptomics, wherein you will learn about single-cell RNA sequencing, how it is generated, how to analyze it, and what specific challenges need to be considered. And at the end of the session, you will have a thorough understanding of marker genes, UMAP and TSNE plots, feature plots, violin plots, ridge plots, and even assign clusters to cells based on observed differentially expressed genes. And these plots are not just useful to understand what the single cell expression is, but they will also add weightage to researchers' findings and publications. Finally, we also have another interesting topic. So let's say we have collected the data on eye color, gender, age, height, and weight of people. We hypothesize that some variables here are related and some are not. And we also think there are exceptions to the rule. The age and height or weight are related, but not always. Relationships observed in data can show which variables are related, unrelated, and if related, then by how much. For example, the weight and height seems to be logically related. Are they in this data? Sorry. Okay. If we plot a trend line, we will find that the trend line goes up, and the higher the height, weight also increases. But the dots are not too close to the line. Some dots seem to be far away from it. And is there a way to quantify this relationship we found? So to help you solve important questions in biology such as these, you will be introduced to 2 more sessions on Introduction to BioML using statistical analysis, as well as Bioinformatics NGS machine learning projects. And as a part of the session, you will be directed to course 7, BioML machine learning for biomedical data, where you will learn about various statistical methods of data analysis, including unsupervised and supervised machine learning that can be applied to biomedical data. And using various examples of omics data, we will see how such analysis can yield significant results for various biomedical applications. And at the end of the session, you will have a thorough understanding of data mining and machine learning, wherein you can perform supervised and unsupervised analysis to analyze patterns, group data objects by feature similarity, and train machine learning models for predictive analysis. So now that we have completed with the coursework, what comes next? In the final session, you will learn how to apply the knowledge gained so far from the coursework to example projects, wherein you will learn about how to download data sets from publicly available domains such as NCBI, GEO, etc. And now for all of that and more, I'd like to pass on the stage to Dr. Raghavendran Lakshminarayan, who will also be the program mentor for this introduction to bioinformatics. So over to you, sir.
Thank you very much, Sri Gauri. Let me set up my screen share. And I think we have a question from a participant. Can you please, unmute yourself and ask your question? We will. Or you can type it in the chat box if you want. But please let me know if you have raised your hand by mistake. Dehumat, what is your question? Please let us know. Okay, there is another question. Yes, Parul, please. I think so. If you have question, please type it in the chat box. We cannot wait longer for people to get your questions done. So we will tend to your questions once you type it in the chat box, or if I'm giving you a couple of minutes chance now. Yeah, great. Okay, you have a question that all the shared details are only about medical or drug designs. Asad, I mean, from my own experience, the issue with plant sciences, right? We cannot, first, we cannot corner everything like only for about medical or drug designs. The development in the omics data analysis in the translational field and medical applications is because we have very well defined analysis setups and pipelines and sequences for completing those analysis with respect to, and also very well defined reference sequences for several of the organisms that we are trying to study, including humans and other bacteria, etc. But I have also worked with a lot of plants genomic sequences. And the issue there is, first, we do not have a complete reference genome for many of the plant sequences. And even if you could find one, then the problem, the complexity of the plant genome sequences actually requires a different setup and a different collection of algorithms and different parameters to be used in a trial and error basis to extract maximum output and maximum interpretation from a plant genomics data, right? So such a method and such a vast human intervention based technique is not viable for our analysis and not viable for many of the resources. We have data regarding or data pertaining to plant sciences where we have, for example, transcriptomics data analysis for plant sequence or plant mRNA sequences, and to understand drought resistance in potato. And we also have identification of contamination in for example, in related to moth sequences and and other plant sequences, right? And we also have metagenomics data analysis of soil, I think soil or rhizobium, I forgot. So those are the data analysis where we have very well defined steps in order to maximize the interpretation and also give you value. If you want to perform other analysis like genomics sequence analysis and this, Asad, you tell me where are the data, where are the reference sequences for the cereal crop, right? Therein lies the complexity. That is why the availability of publicly available resources, right? That is why we even if we want to perform that, all of these analysis will need specific curated analysis sequences, pipelines need to be set up for several of that. And even if we could present you a whole genomics data analysis for one specific plant genome, that might not be the same steps or same setups and parameters that you are going to apply for another plant genome. Then we cannot generalize. If we cannot generalize, then we cannot prepare generalized material. That is why we have limited resources on plant science. Not only us, you take any available publications, right? They have different methods pertaining to plant genome sequences. That's why. But wherever it is available, we are trying to expand our resources to include plant data also. Okay. Yeah. So, okay, coming back to your questions, right? Asad wants to do understand about single cell transcriptomics of cereal crops, right? So we can break this down further, right? If your focus is on understanding the analysis of single cell transcriptomics data, we have plenty of resources on that, right? You can set aside the system and understand the process, and maybe by that way you can adapt this process to suit your system. that is how you can proceed. If you want my interpretation about how can I proceed with given this condition. And Dr. Rita wants to do analysis of maize against fungal diseases. It is actually possible to do if you have reference sequence for your maize species. We have please mute yourself. And we have a very closely related project, exemplar project, which I might not be focusing on discussing today, but we have it in the Learn portal about how to map the sequences to a known reference sequence, and then take the unmapped reads and then try to identify from which fungal or viral source that these unmapped reads are going to come from. So your question, Dr. Rita, is very similar to this. This method will work only if you have a good reference sequence, genome sequence for your maize species. So that is a must. If you do not have, then it's going to be very difficult to participate in this analysis, because work with this analysis, because you have to do de novo genomics data analysis, where the de novo analysis platform or algorithm will also take your fungal sequences and start building up contigs and contigs for that, okay, and assemblies for that. So that is why. Yes, we are going to talk about that in my session. So Parul, please wait for that. So let's go over. Let's go over what we have in store for trying to apply what you would have learned so far when going through this program, right? The program is going to teach you, our program is going to give you a general overview, a great overview of what are the different analysis options you have, and what are the different setup options for those analysis algorithms, and when you should use what algorithms, and in what combination you will use these algorithms to study what, right? To give you a more practical approach of all of these or several of these analysis options, we have prepared several of these exemplar projects in the Learn portal. So what are these exemplar projects? These exemplar projects will take data from publicly available resources, from a publication or from public repositories, and then start asking relevant biological questions to these data, right? If it is from a publication, we are going to ask a slightly different question than what is being addressed in that publication. And then just like how to approach a research project, we will approach that question by setting up methods, and then we will perform those analysis with the resources that we have, and then we will interpret the results, and then we will implicate them biologically at the end and compare it with the original publication, or compare it with the publication that refers to these data generally. This is the structure of several of the exemplar projects that we have, and for some of them we are going to, or I am going to give you an introduction how what you will be learning throughout the course of this program, right? So if I hope my screen share is working. So the applications of bioinformatics is very well explained by Sri Gauri. So if you are an absolute beginner, so, and Sri Gauri also mentioned this, I would want you to start with these getting started courses. Like you can start with Introduction to Bioinformatics, you can start with Bytes and Molecules. If you want to expand your horizons or expand your knowledge into scripting world, you can start with Bioinformatics in R and Bioinformatics in Python, although it is not absolutely necessary to learn scripting to perform bioinformatics, as we are going to present to you. You can use the resources as we will demonstrate you how to use the big data bioinformatics resource of Taobao Bioinfo platform to perform all the necessary analysis and understand the biology behind this data analysis, big data bioinformatics, right? And after this, you will be able to see, or after this program or throughout the course of this program, you will be able to see how NGS data and bioinformatics are used in various research areas that includes neuroscience, agriculture, infectious diseases, oncology, and etc. So as discussed earlier also, we have several examples on some of these topics and a few examples on the other topics. So if you want to get creative on applying one method to another system, so you are very welcome to do that, and that is how active research is being carried out as well. So let's start this with trying to understand how the course genomics, or the coursework, or the resources that we find in genomics can be applied to a practical example, right? This course is going to give you a complete overview of what are the different analysis that you can set up for genomics data analysis, right, as mentioned, right? And how to choose specific analysis type for specific question is what is answered in several of the example projects. Some of the projects that are associated with genomics data analysis is to understand the SARS-CoV-2 variants from wastewater surveillance. That's an excellent project. And for example, to do a comparative analysis of phylogenetic tree analysis and evolutionary reconstruction of zoonotic transmission of SARS-CoV-2 variants or SARS-CoV-2 virus from species to species in a comparative whole genome sequence analysis. That's also that. But what we are going to focus here is to understand, for example, by the use of mutational analysis, how drug resistance in pathogens are going to be developed. So we know about Mycobacterium tuberculosis, and we know the background of tuberculosis and how Mycobacterium tuberculosis is very, very important to study, being that is the leading cause of death in several of these countries apart from cancer, right? And one of the main problems with dealing with Mycobacterium tuberculosis pathogen is it can, for example, develop resistance to most of the antibiotic that it gets, or that it is that the patients are treated with. So this also gives us a, for example, a general complication in not only in tuberculosis, also in other pathogens, on how these pathogens develop this drug resistance, and can we from excuse me, from genomics data and analysis, can we pinpoint the driver genes or driver mutations that are responsible for the drug resistance of these pathogens, right? Yeah. Mm. Mm. And the increasing prevalence of these drug resistance forms of TB infections, for example, obviously requires more effective and more personalized treatment strategies, right? But the problem is the current standard regimen actually includes combinations of different antibiotics or combination of different types of antibiotics for a longer period of time. And all of these current regimen, treatment regimen, are prone to develop drug resistance in the pathogen, right? Uh, the ideal scenario for the pathogens to develop drug resistance is prolonged exposure to the antibiotic. So that is definitely in this regimen. And prolonged exposure to multiple types of antibiotics, that is also in this regimen. So we have to strike a balance between treating the pathogen and also not letting the pathogen develop resistance to these drug. But in between these analysis, if we can understand how this drug resistance is actually being developed, we can target specifically to those pathogens and then try to eliminate those pathogens from the infectious person, right? So this is to show that how complicated this drug resistance can get. For example, over the course of this drug resistance, development of drug resistance in the pathogens can actually develop resistance to multiple drugs, which is called multi-drug resistant tuberculosis, or extensively XDR, extensive drug resistance. It is being clocked, or it's been counted that multiple drug resistant tuberculosis can be resistant to almost 38 or 39 different drugs, right? And extensive drug resistant tuberculosis can be resistant to up to 71 different drugs. So both of these are extremely dangerous, and we do not have effective plans to treat these drug resistant Mycobacterium tuberculosis pathogen. So how are these actually generated, or how are these drug resistant actually is developed? So there are generally 4 groups of anti-tuberculosis drugs, and the classification is actually based on the mechanism of action of these drugs, right? And whenever there is a drug mechanism involved, and whenever there is a gene that is involved in this drug mechanism, then the genes that are targeted in the pathogenic system also has a susceptibility to develop mutations that can resist this targeting mechanisms. So that is why stochastic mutations targeting these associated genes can lead to resistant to those specific drugs that are targeting these particular genes. So this information also gives us a window to probe these genes for known, characterized, and also unknown and uncharacterized variants and mutations that you can identify from performing a straightforward genomic data analysis or variant calling pipeline analysis. That is what we are going to do in this specific project. So this specific project deals with this publication, where this is a wonderful publication where it it tracks the drug resistant development of one pathogen in a single patient for 21 years. So over the course of this 21 years, the patient was given different treatment regimes, and then the development of the drug resistant to different drugs and different antibiotics is what is recorded. And then we have a window, or we have a unique opportunity to take the isolates at different time points of this one specific MDR variant, and to understand how different mutations and how different genomic variants can lead to development of resistant to several of these listed antibiotics that this pathogen came into acquaintance with, right? So to perform this analysis, we use straightforward variant calling analysis, right? We map the sequences, or we map the isolates reads onto the reference sequence of Mycobacterium tuberculosis, and then we visualize for visual confirmation, and then we perform variant calling pipeline. There are different of these variant calling pipelines. Some of them will work better than the other. So it's always very helpful to try more than one. So it is often done more than one type of variant calling algorithm is actually used, and then sometimes the consensus is actually used for downstream analysis, or the the union of these variants identified are actually used for downstream analysis. So that's what we will actually do. And what you will actually gain is to understand and identify what is the variant that is statistically relevant, right? From visualizing these variants from BAM file and VCF file, and to, for example, identify those relevant or key variant that we want to focus on that could be responsible for the development of these drug resistance in this Mycobacterium tuberculosis, right? And that is what we are going to do in this specific project. So if you are interested in trying out this project before proceeding, so you are extremely free to check this out, right? Okay. So what do we have here? Okay, this is this we don't want to focus now. Okay, so all the information and all the analysis steps to perform this analysis, and the interpretation to how to interpret the outputs and how to compare it with what was actually published is actually given in this project. Thanks, Sri Gauri, for pasting the project link. So that is how you understand how to set up, for example, how to use a variant calling pipeline analysis of genomics data to understand multiple drug resistant in Mycobacterium tuberculosis. So this gives you an opportunity to take this analysis and modify it for your needs, right? The data does not have to come from this one particular resource. You can find completely freely available multiple drug resistant publicly available data from NCBI or from other publication, and then try to repeat this analysis and find the difference between the variants that you identify for your data and what we have identified as per described in this specific publication. That's why you start learning how to apply these analysis methods to a variety of different data. So let's move ahead and discuss about what can we learn from metagenomics data analysis, and how, for example, applying metagenomics data analysis to one specific project will help us gain a deeper understanding of the general overview of metagenomics data analysis. So again, what the course contains, what are the contents of the course, and what are the contents of the projects and how they differ is already been discussed in the previous set. So this also follows the same. The course is going to give you a complete overview of all different possible analysis options that you can perform for different data generated from different systems. The data can come from human samples, right? Like human American gut microbiome project, or data can come from a model system, which we are going to discuss today, right? That is also possible, right? And what are the, for example, patterns in the data when you are considering data from a model system or when you are considering data from human resources is what will be that determines the interpretability of your risks. So generally, for any system, the control that the model system gives you also helps you to increase the signal to noise ratio. And the lack of control that one has to encounter with the human data is actually associated with more noise that we want to eliminate or that we want to suppress during the course of our data analysis. So the methods to do that and where to focus on which part of analysis to focus for what model system will be the main takeaway from this specific course, right? So let's focus on one particular aspect, right? So this is already been discussed and we already know, right? In healthy adults, gut microbiome, the microbiome that is existing inside our human gut, it actually exists in a state of mutual symbiosis with us, right? The host, right? the environment of this symbiosis or this gut actually dictates both the composition of these microbes and the functional productivity of this microbiota, right? And this microbiota has been known to interact with the host through various ligands and various molecules that also forms many key important metabolite that we have to rely on to process the diet, process the food, or to generate energy, or for immune response, and they are implicated in several of different biological processes from our body itself, right? So due to this interaction, we now have an understanding that gut microbiome has an enormous impact on the life of host. And what impacts or what has more impact on this gut microbiome itself is the diet that we take. Right? The diet that we take plays a fundamental role in shaping the composition of this microbiome and, and this function. And hence, this also leads to, for example, this also leads to how the food is actually processed and how much energy that we are actually extracting and how it generally affects our other bodily functions due to the balance or imbalance in the gut microbiome that is present in our body. Right? With that background, let's move on to this specific project. So this specific project is it deals with the role of high-fat diet, right? To understand how high-fat diet is going to shape the gut microbiota, the system that we are going to deal here is the mouse model system, right? A model system gives you more flexibility of completely eradicating all the microbiota that is present in the gut of this mouse or these mice, and then start rebuilding those microbiota based on the diet that we are giving these mice. Based on the diet regimes that we are giving these or providing these mice, right? So there are two types of mice that we are going to deal with. One is given a normal diet, which is chow diet, and the other mice is given high fat diet. So we are going to find out the imbalance or symbiosis or dysbiosis relationship between the microbiota and their dietary components, right? This publication is wonderful in essence because it has a lot of layers. It not only studies the imbalance that is generated by the diet, right? It also studies the behavioral effect of these microbiota symbiosis that is generated due to the dietary supplements, right? So for this project, we will show you how to set up amplicon sequencing analysis of amplicon sequencing data using data to pipeline, and then we will show you how to interpret the results from, let's say, a proportional abundance plot or abundance plot of different regulatory diets. So in this, for example, data, if you check, where is yes, for example, in this data, you can check that there are several of the bacteria that are abundant in high fat diet compared to the regular diet. For example, the phylum Saccharobacteria and phylum, for example, Actinobacteria are abundant in high fat diet, but they are completely missing in these. So that is what we will learn. And also we will learn to identify those specific bacterial phyla or genus or species that one can identify being particularly or specifically abundant in one type of phenotypical conditions or sample conditions, which here is a high fat diet or chow diet. So we see that the composition of, for example, bacteroidetes versus firmicutes changes depending upon how many samples that we are depending upon what these samples are actually, where the samples are actually collected from, right? And furthermore, this project will also show you how to perform a grouped abundance analysis like alpha diversity and beta diversity, which can help you summarize the diversity of population in each of these phyla or members that belongs to these different dietary samples or samples from different dietary regimes, right? Um, we can learn what the alpha diversity shows us, right? How we can estimate the number of species, or how even the species spread is between the samples, right? That's what we can learn from the alpha diversity. And we can see what differentiates, right? How the samples are differentiated based on the amount of diversity present within them and between them by analyzing the beta diversity. So this is what we will get to learn in this project. So what do we understand by trying to learn or going through the process of performing this analysis and trying to relate these difference in diversity and difference in abundance to different populations, or to different sample conditions or different dietary samples with different dietary regimes, is we can, for example, uh, take or we can measure the level of symbiosis or dysbiosis within the system as a function of these abundance of different microbial populations at the rank that you chose to. If you want to understand the population based on based at the phyla level, we can do that and implicate that to different disease cases, right? And if you want to understand the population with a bit more resolution, we can go up to genus level and understand their composition also. So there are certain limitations in going beyond the genus level, as you will learn throughout the course of this project, that where these level ends and what to do if you encounter a lot of uncharacterized microbial population, right? So in the end, the aim is to relate the abundance of these microbiota to, for example, to understand the change in their balance from symbiosis condition to the dysbiotic condition, and relate that to implicate that to different dietary conditions or different phenotypical conditions of the data or different disease state, if we are going to focus on that. So this project focuses on understanding the dysbiotic development of microbial population due to the role of high fat diet in the samples. So that is where we are going to focus this. So I want to stop at this point and ask if you have any questions. So Srigori, if you can paste the link for this particular project, that will be great. And if you have, yeah, thank you very much. If you have any questions, please put that up in the chat, and then we will quickly go through what we will learn in the next few project examples, right? I want to rush this a little bit because we are short of time. So the next section or sessions that you are going to encounter is transcriptomics data analysis, and transcriptomics course itself is a huge course, and it has lots and lots of options to perform how these transcriptomics data analysis can be performed through different combinations of analysis methods, right? I don't want to go over everything because we have project example that addresses each and every one of these combinations. If you want to, for example, map this onto the genome, we have a project example for that. If you want to map the reads onto the, for example, isoforms to get isoform expression level, we have a project on that. If you want to build your own transcripts, we have a project on that. So please go and explore different project examples for transcriptomics data analysis. So that is going to be very helpful, because transcriptomics data is the most used data in the spectrum of omics data for implicating in several of diseases or biological conditions or phenotypical conditions, and to understand generally disease progression, development of mutation, development of cancer, and a lot of other implications also, right? So what are we going to focus on today? So today the focus is on understanding the transcriptomics landscape of Alzheimer's disease, and try to find some relevant pathways that can be implicated in the disease biology or disease progression of Alzheimer's disease. Right? I don't want to go over the background of Alzheimer's disease. Everybody knows that how progressively neurodegenerative this disorder or disease can be, and people actually usually start from suffering from loss of memory and cognitive functions, and also eventually leading to the loss of ability to respond to environment and even to perform daily tasks. Right? So let's go over. We will deal with how to prepare data for example, to understand different ways of data pre-processing for exploratory data analysis like PCA, or performing quantile normalization or histogram analysis, and what these analyses are going to show us or tell us. Right? We are looking for example, here we are looking for good separation between samples from Alzheimer's disease and sample from control disease. So what you can learn from this PCA or exploratory data analysis, I can learn that there is, in addition to these samples being separated based on the disease state or control, there is also a decent level of separation between some of the samples within these disease levels. Right? They might affect my data analysis or they might not. So it depends upon, I can check at the phenotypical characteristics of the data and understand their separation, or I can just proceed forward. Right? It is safe to proceed in this case. Why? Because of the tidbits that we will discuss in this session, like the scale of the principal component axis here and here does not warrant a special investigation of why these samples are far apart. Right? The scale is extremely minimal, and it is okay if they are a little bit far apart. So that is not going to affect our data analysis. So we proceed with performing differential gene expression analysis to understand genes that are differentially expressed, or that are upregulated or downregulated. And we can perform some very well-known analysis like volcano plots and LFC plot, MA plot, and etc. And and identify pathways that are associated with these differentially expressed genes and pathway association analysis. Right? By performing these pathway analysis and identifying pathways associated in the gene ontology terms or in the KEGG pathway repositories, we can, for example, understand what are the different enriched pathways or suppressed pathways that can be implicated in the disease progression of this Alzheimer's disease case. Right? That is what our ultimate aim to. Right? When you perform these analysis as described in this project, eventually you will try to, or you will come to a understanding that several of the synaptic pathways are impaired in the Alzheimer's disease patients, and several of the other pathways are actually suppressed or activated, like immune response and other pathways are activated in the Alzheimer's disease patients, and we can find biological reasoning or implications for these pathways based on the disease biology. So this excuse me, this and several other examples of what to learn from performing a simple, straightforward transcriptomics landscape analysis based on differential gene expression analysis for Alzheimer's disease pathology is given in this project. So please go through if you are interested in this. This is just one example project that is based on transcriptomics data analysis. We have several projects that focuses on different sections or different applications of transcriptomics data analysis, like in understanding the drought resistance, to understanding the cancer biology, right? Understanding the host response to SARS, right? Understanding the effectiveness of drug response in cancer, right? Cancer cell lines. So there are several of these applications, and we want to showcase. And finally, right, this is an interesting project and excellent project if you want to learn about how a sample data can be or how, for example, cell lines data that is recorded for multiple omics characterization and their drug response to several different therapeutic options for breast cancer being studied. And we can perform in this project, we show you how to use Taubot Bioinfo platform to perform unsupervised data analysis, unsupervised machine learning analysis, and supervised machine learning analysis on this data to understand, for example, breast cancer subtypes, and to use multi-omics integration to understand drug response to 70 different drugs that is tried on these cell lines, right? That is what this particular project deals with, and all of this you can do without coding, right? The aim is to make you understand the logic behind this analysis, and if you are interested in expanding your knowledge to include coding, we have same analysis or same projects done on R or Python's platform also. So with that, I will conclude my session and pass the stage on to Sonalika, and I am free to take some questions if you have any, and I look forward to meet all of you over the course of this program. Over to you, Sonalika, and thank you, Srigauri, for pasting all of the links for the projects, and they are very useful. Thank you.
Thank you so much, Dr. Raghav. Right. Srigauri, is my screen visible?
Yes, Sonalika.
Thank you. So I would like to now take you all through the program page of the program Introduction to Bioinformatics. And on this program page, you will find the schedule of the program, on which all dates we have different sessions scheduled, and what all topics are we going to cover for each and every session. And along with the session topics, you also have some associated online resources, which are the lessons from different courseworks on the Omics Logic Learn portal. These resources are hyperlinked, so if you just click on the specific resource, you'll be redirected to the specific lesson of the course that you have to take up as a part of this session. Now, this is the schedule that you can go through. One more thing, on the program page itself, you'll find the Zoom link when you have to connect for the specific session on the program page itself. We will also email you the Zoom link that you have to join for each one of these sessions. And once the session is over and the recording is processed, we'll upload the recording of the session on the program page itself. So let's say you're not able to join the live session because of some priorities, then you can watch the recording of the session and come back to us later with any questions that you might have. Now, I like to open up another program page just to give you an example how the recordings are put up on the program page that the participants can access after the program. Also, you can revisit the topics that were covered during the session. Once the session is complete, once you have watched the recording and once you have completed the resources, you need to click on the complete session button like this. I'm taking an example of another program right now because Multi-Omics Introduction to Bioinformatics is yet to begin. So the program page is not yet active. And once you click on the complete session like this for all of these sessions, once you have completed this for all the sessions, you will be able to get your certificate in the profile tab itself. So that was about the schedule, where you can find it, where the resources are listed, what topics will we cover, and also how you can find the recording of the session on the program page itself. Now, the next thing is for those participants who are yet to enroll themselves for the program. If you scroll down on the program page itself, I just put the link. All right. Thank you so much, Srigauri, for putting the link of the program page in the chat. So the link is in the chat. Now, once you scroll down, you can find 3 different levels of subscriptions that you can pick to enroll yourself for the program. The first one is the 45 days, and then you have 60 and 90 days. Now, the duration of the training program remains 45 days. That is, all the sessions, all the training material would be covered in the 45 days itself. What additional do you get if you enroll for 60 or 90 days is the chance to work on a research project and get it published. So that is the difference, and that is the additional thing that comes apart from the training. So once your training is complete, you are supposed to submit a research proposal to us after the training, and then we start working on your research project. Everything would be under mentor guidance, and then you complete your project and submit it. So these are the different pricing that is mentioned for all these different subscription levels. Now, we are offering partial scholarship, that is 50% off on all of these different subscription levels. Shubham will just be sharing the link or the coupon code. The coupon code is there in the chat box. It's multi-50. You need to click on the buy now button if you want to enroll for the program and put the coupon code here. Once you put the coupon code, you will get a partial scholarship for the program, and then you can proceed via Stripe or PayPal. If you're not able to proceed with these payment options, then you can reach out to our team on marketing@omixlogic.com. They'll be able to share with you a payment link or the other options as well that you can choose from to enroll yourself for the program. So that is how you check out for the subscription level that you want to pick and enroll for the program. And if you face any issues, feel free to reach out to us. Shri Gauri has shared the details in the chat box already. Our team will be there to help you enroll yourself and get started. So now here you can see that the pricing is mentioned in dollars. Now let's say if you are from a different country and have a different currency, you can go on top and click on the specific currency that belongs to you. Now I'm in India, for me it's INR. So I'll just select INR and then the pricing would be changed to INR. And the same process, you apply the coupon code and then you can see the partial amount through which you have to proceed further. So that was about the 3 different subscription levels and what additional do you get. So in the 60 days, you'll be able to work on a research proposal and the outcome could be a poster presentation. And then in the 90 days, you could work on a research project itself, again with one-on-one mentor sessions, and you will get a research license on the T-Bioinfo server where users can upload their data and then run on the pipelines. So that is the difference in 3 subscription levels. Now, if there are any questions, you can please put in the chat box so that we can answer regarding subscriptions or scholarships. Yes, definitely. So as I mentioned that once you complete the training program, then you can work on a research project. And it's not that you have to work on a research project that we offer you, like a topic that we offer you. If you have a research proposal and if you want to work on your research question, you can definitely propose it to the mentor and then start working on it. Azad, this was for you. Yes. Any other questions? So if you're focusing on sorghum, we want to know what type of research question you want to answer related to this specific plant. And then if you have a shortlisted data set for yourself, perfect. If not, then throughout the training, you'll be also taught how to extract data set. So you can then select a specific data set and you have to provide a background of your research question. So we have a research proposal form that we generally give to our participants. So we'll give you a research proposal form where you have to mention your research question, the background of your research, whatever literature review you have conducted and what exactly you want to answer, the link to your data set, and some of the references that you went through and referring which you want to work on the question. That's it. You need to prepare a few presentations. Once you complete your training program, we will have an orientation session with you where we will explain everything, how you have to start with your research question. So there we generally tell that how you have to prepare your presentation of the research proposal and present it to your mentor. The mentor will then let you know if any modifications are required. If there are, then you can meet them and get started with your project. All right, perfect, Azad. I'll share the link of the research proposal form as well. Please fill in the details so that we can discuss on your topic. And Azad, please put your email address in the chat so that we can have a one-on-one call with you to discuss the specific research topic that you want to work on, and then we can get started. Thank you, Azad. We have a question by Nashmia. What is the course start date? The course commences on 23rd January. Price in euros. Yes, I can. If you go on top and scroll here and you select the euro currency, then you can find the pricing here. 149 euros for 45 days, 299 for 60 days, and 499 for 90 days. But again, there is a partial scholarship that we are offering. So if you put the code, it would be half of the amounts that are mentioned here for each one of these subscription levels. Nashmia, this was for Estella. Ujasvi has again put the coupon code. Thank you so much for sharing. It's multi-50. And also we will be sending out an email with the coupon code that you can use. If you copy the coupon code right now, perfect. Otherwise, you'll also get via email. And those who are interested to enroll themselves with the partial scholarship, they can put their email address in the chat box so that our representatives can help you get started with your journey. Yes, definitely, Nashmia. You can shift from 45 days to 90 days after completing the 45 days of the training time period. All right. Are there any more questions? You can still ask. Our team is here. We can assist you. All right. So, Estella, the code is for everyone. For any participant who wants to enroll for the program, they can use the code. They can be undergraduate students, postgraduate students, PhD scholars, researchers, anyone from non-bioinfo field, from bioinfo, anyone who wants to enroll. Estella, please share your email address so that the team can reach out to you with the details. Thank you. All right. So if we have no more questions, I think we are good to end today's session. And again, if you have any further questions that you want to ask maybe later or via an email, please feel free to mail us at marketing@omicslogic.com and we'll be glad to assist you. Thank you everyone for joining today's session. It was nice interacting with you all. We'll meet you in the program. Thank you. Bye-bye.
Thank you, everyone.